right. So, like I said, we won't keep score because there's only a couple of you. So you can. So after class is over, I'm going to post at the top of the Word document. It has the links to these 2 Jeopardy boards. So there's this one that's 5 columns, and then there's a second board that only has 2. You have to pay money to have more than 5 columns, so that's why I made 2 tabs. So in terms of the structure of the test on Monday, so it is 61 questions. So there are 30 questions on the chapters 6, 11, and 12. So those chapters are bones, fundamentals of the nervous system, and then the central nervous system. So half the test is that. And then there's another 10 questions from the bank on the hip and knee. So that gets us up to 40. There's 10 questions from test 1 from that test bank. So that's mostly muscles, but there's some other stuff in there. And then there are 5 questions from the elbow down, and then 5 questions on the shoulder girdle. So rolling cumulative. And then there's one big 5-point question, true or false. All right, so let's just run through these. So Connor, since you're on my left, do you want to go first? Pick a category. All right. So describe concentric, eccentric, and isometric muscle contractions. All right. Jack, you got anything on that one? So close. So overcoming the resistance. So whatever you're able to contract the muscle more, so the muscle is shortening, and typically you're going to get a decrease in joint angle. So that's concentric muscle shortening. Eccentric contractions, we are slowing down most of the time, so it's usually on a follow through. And then on isometrics, that's going to be where the muscle is contracting, but there's no change in length. So we see that, we talked about stabilizers, but you also see that when you're pushing against immovable objects, like a wall or something like that. Isometric contractions, no change in muscle length. All right. Cool. Jack, what are you going with? Bones for 100. Starting low stakes. All right, so what's the name of the double-layered membrane that covers the outer surface of bones? It is the periosteum. Good, yes. The periosteum is on the outside. That's 2 layers, the osteogenic layer and the fibrous layer. Remember the osteogenic layer is where we lay down new bone, osteogenic, start of bone. And then the fibrous layer is the layer that the tendon hook into. Good. All right, so we got that one. Back to Connor. 1 for 2 so far. Okay, there we go. Name 3 muscles that insert along the leg. So 2 of the quad group originate along there. So the group of muscles that insert there includes muscles that have M, L, and B, if that helps you. Jack, do you remember? So the group of muscles that inserts there are the adductor muscles. So adductor magnus, longus, and brevis, that's where the MLB comes from, as you can see my sport orientation. How I remember. And so any of the muscles that insert along there, then you've got adductor magnus, adductor longus, adductor brevis, all 3 insert. So those are the ones I was looking for, the adductor group. All right, back to Jack. Yes, let's go big. Bones for 5. Ooh, that's a tough one too. All right, so what are the 5 zones of an epiphyseal plate, and what happens in each? Like that's a thing, but it's not taking up brain space anymore. All right. Connor, remember? And you can use your notes, So the 5 basic zones of so remember epiphyseal plate, what's the more common name for that? That's a growth plate, right? Yes, it's a growth plate in a long bone. Yeah, and so in terms of the zones, so we're starting near the epiphysis, near the rounded end of the long bone. So the first zone is the resting or quiescent zone, so basically nothing is happening. Then we have the proliferation zone. Remember, if something proliferates, we make themselves. So that's the proliferation zone. Then they grow up, which is the next zone. That's the hypertrophic zone. So first they split, then they get bigger. And then you get calcification of the matrix. Sort of supply lines. They cut off their access to the nutrients that they need, oxygen, carbohydrates, amino acids, etc. And so then the chondrocytes die. And so then what you're left with is sort of this spider web of the zone. And then your bone cells move in there. So the osteoclasts and osteoblasts migrate to the area, and then that becomes the osteogenic zone. So then you actually turn it into bone. So the 5 zones then, quiet zone, the resting or quiescent zone is the way the book phrases it. So resting or quiescent zone, hypertrophic, calcification, ossification. So we calcify it, and make it into bone. All right. And then back to Connor. Sure. Dealer's choice. All right, let's go fundamentals of the nervous system for 5. All right, describe the effect of IPSP membrane potential of a postsynaptic neuron. So there's a lot of terminology in there. Part of the question is I want to make, or part of the thing I put this on here is I want to make sure that you understand the terminology, and then we'll talk about what it means. So this is pretty recent. This is at the end of fundamentals. This is day 2 of fundamentals. I don't know if that helps, but. Okay, good. Yeah, good. So that's the basic information. Excellent. So let's start here. So, got a ladybug. So IPSP stand for inhibitory postsynaptic potential. So they're going to inhibit that receiving neuron. So that means that they're going to make it harder for it to send an electrical signal. EPSP is excitatory postsynaptic potential for that postsynaptic neuron to send an electrical signal. And how they do that is they change the resting charge of the receiving or postsynaptic neuron. So if we send an inhibitory signal, making it harder for it to fire, what we're going to do is make the inside more negative. So it'll move from negative 70 millivolts at rest to negative 75. And so physically the way that we do that is we allow some potassium to leak out, because potassium is a positive ion, and there's a lot of it. Allow some of the positive ions like potassium to leak out, make it more negative, or allow negative ions like chloride to come into the neuron. And ions to come from negative 70 to negative 65. So it gets it closer to that threshold voltage of negative 55, or sorry, where the electrically gated channels start to open. So if you remember nothing else, IPSPs, inhibitory, make the inside of the cell more negative. EPSPs, excitatory, make the inside of the cell more positive. All right, Jack, what do you want to go with? All right, in what lobe of the cerebral cortex are the visual and association areas? So where is visual input processed in the brain? What's that? Yeah, it's occipital. Good. Yep. So the right. Yeah, that's in the frontal lobe. You're using lobe. All right, that's pretty good. What do you think, Connor? All right, this hormone is released in response to low blood calcium levels. It stimulates the osteoclasts to resorb bone, which means to break it down, releasing calcium into the blood until blood calcium levels return to normal. Once calcium is normal, this hormone stops being secreted. So what hormone causes the osteoclasts to break down bone to release calcium from it? Keep our blood calcium levels within normal limits if they fall too low. Jack, any ideas? Parathyroid hormone is what I'm fishing for here. So parathyroid hormone secreted from a gland in the neck. And so that is going to stimulate the osteoclast to break down bone and release calcium. So parathyroid hormone or PTH. All right. Jack? Name 5 actions for the sartorius muscle. Or across the 2 joints. Yeah. Yeah. Okay, internal rotation at which joint? It is one at one joint, but not at the other. What's that? Yep, it's an internal rotator at the knee, so tibiofemoral. Okay, that's one. It is a flexor at both joints. Yeah, so it's a tibiofemoral or knee flexor. Acetabular, 3 actions. So remember, sartorius does all the stuff when you cross one leg over the other. So we got flexion, flexion, and then internal rotation. But I'm doing 2 more things in my hip that I haven't named yet. Deduction. Doing at the hip when I cross one leg over the other. Yeah, internal knee. The femoral. Deduction. AF. External rotation. AF. Internal rotation. All right. Connor. Okay. Name 5 muscles that are agonists for flexion at the acetabular hip joint. Sartorius was one. Not according to the anatomy. One, but not examples. But ones include tensor fascia lata, TFL. Pectineus is another one. Adductor magnus, adductor longus. That makes it the big 3. Iliacus psoas or iliopsoas, and then rectus femoris. All right, Jack. Which one for 3? Oh, okay. Yep. All right. Describe what happens to the brain in a concussion. Okay. So the 5-point question, in case anybody actually watches the video, is this question that says something to the effect of, in order to have a concussion, the brain has to strike the skull. It is false. The brain does not have to make contact with the skull. The brain gets accelerated or decelerated. And either axons of the neurons in the brain. When you reach out, like a calcium come inside, and so that throws off their normal firing. They're not able to. If you get enough damage, when you get a loss of consciousness. So particularly if you get damage in that frontal lobe of the prefrontal cortex, that's just a stretching or compressing of the axons of neurons, and so they can't maintain their electrochemical gradient, they can't send electrical signals, and so they are essentially non-functional for a little while. And so while they're non-functional, they can't do their jobs, so then that becomes manifest as either loss of consciousness or memory, the typical stretching or compression of the axons of neurons. All right. Connor? Okay. All right. If someone suffered damage to the auditory association area, how might you know? So yeah, that will work. Yeah, so that's an association thing. So the gist of it is, for your primary auditory, you're going to area. So if there's damage to their association area, they can't make meaning from sounds. They can't put together, beats and guitar noises. Because Wernicke's same part of the brain. That might also extend to they can't understand speech or directions. So they can't make meaning from sound. All right, a blank is the structural unit of compact bone. It's comprised of a group of hollow tubes increasing in diameter, one outside of the bone. And it starts with an O, if that's helpful. It may not be. Yep. It's been a while since we did bones. So actually, I forgot it was part of this test until like last week. I was like, oh, I got to come up with some bone questions. Is what I'm fishing for here. Yeah, right. It's bone something. So in this case, it's an osteon. So it's that series of rings around each other, and then so it's that cylinder, and then you have through the middle of it. All right, Connor, what do you think? Oops. What are the 3 layers of fascia in a muscle? So 3 layers, they're all mycium something, right? So you got epimycium, wraps around the entire muscle. Yeah, so superficial to deep. So paramycium wraps around a fascicle or bundle of muscle fibers. And then endomycium is the layer of connective tissue around a muscle fiber or muscle cell. So epi, peri, endomycium. Bones and bones. All right. What is an epiphyseal plate? Where is it found in the long bone? Okay. So growth plate, yes. So remember there's 3 primary parts to a long bone. So you got the epiphysis, and metaphysis. So in which of those 3 is the epiphyseal plate found? So the diaphysis is the shaft. So it's not there. Yeah. Yep. So the three parts of the long bone, diaphysis is the shaft, epiphyses, or an epiphysis, is the end of the long bone, right? The growth plate is actually found in the metaphysis, which is the area, the widened area between the epiphysis and the diaphysis. So on this one, it makes sense, right, that the epiphyseal plate would be in the epiphysis, and so that's obviously one of the multiple choice options. Don't pick it. So it's in the metaphysis. So it's in the widened area and not actually at the end of the long bone. All right, Connor, what do you think? All right, what are the three components of the blood barrier? And what is the function of this barrier? It's very Yep. Good. So the three components are the continuous endothelium. What does that mean? The inside of a blood vessel. So in the case in the brain, those are continuous. There's no gap between those cells. So you got a continuous endothelium. And then you also have a thick basal lamina. What's that? That's the connective tissue around the blood vessel. And so the harder it is for things to diffuse across it, because in order for something one side of a barrier, the harder it's going to be. So there's a thick basal lamina. And then you also have, so remember the astrocytes are glial cells, they're helper cells in the brain, and they have projections that actually wrap around the blood vessels. So between those three, it's tough for things to move from the bloodstream into the brain. Why is that? Serves a protective function. So we don't want things like viruses and bacteria able to migrate from the bloodstream into the brain, because again, neurons are amitotic. We can't just make a new one if that one dies, as opposed to, if cells in the small intestine die after you get a stomach infection or intestinal infection, we can just make new ones. We turn those over all the time. It's not a big deal. But if we get an infection in the brain, well, then that, obviously can shut down the heart. That can kill us. And so we need to make sure as few things as possible migrate from the blood into the brain. So that's the function of that barrier. All right, to Jack. What are the three parts of the diencephalon, and briefly describe each? So the diencephalon, on the picture slide textbook, and the diencephalon is always purple on there. Brainstem's green, diencephalon's purple. So the three components are the thalamus, the hypothalamus, and the epithalamus. So they'll have thalamus in them. So what does the thalamus do? Do you remember the one thing I wanted to make sure you remember about thalamus? Information comes back, a lot of it synapses in the thalamus first and then goes elsewhere. So then maybe goes from the thalamus to the somatosensory cortex, goes from the thalamus to the auditory areas, etc. And motor information similarly can go down through the thalamus, or there's kind of we talked about with the basal ganglia and the cerebellum that go through the basal ganglia and then information and output information. So know the thalamus is the relay center. What about the hypothalamus? What's the one bullet point I want you to remember about it? Controls the autonomic nervous system. So it exerts influence over our core temperature. So it initiates shivering or sweating, plays a role in satiety, plays a role in making us hang on to water. Like if we start to get dehydrated, it stimulates the release of antidiuretic hormones, so we hang on to fluids. So it keeps us in homeostasis. Doesn't let us get too hot, too cold, too dehydrated, etc. Yep. So typically you have to take diuretics. Fluid balance. If your kidneys aren't functioning right, the more fluid you have, failure. Pressure down by reducing. Yeah, good question. And then the last part's the epithalamus. And so the epithalamus, the only thing about it, I won't actually ask you about it, but it controls sleep and wake. So it plays a role in that. And so does the hypothalamus, for what it's worth. So thalamus, hypothalamus, epithalamus. All right. To Connor. All right. What are the 4 muscles of the quad group? Nice. So the 3 vastus muscles, so vastus medialis, vastus lateralis, vastus intermedius, and then rectus femoris is the last one. Good. All right. All right. How does the nervous system code for the strength of a stimulus? So for example, how do we know if I'm stretched quickly rather than slowly? That's a good guess, yeah. So from the neuron standpoint, the more intense the stimulus, the so back to the muscle chapter, we talked per second, greater frequency of depolarization. So we want just a weak muscle contraction, it'll send 20, 30 action potentials per second. If we want a strong muscle contraction, it's more like 80 action potentials per second. So the way the central nervous system codes for the strength of a stimulus is based on frequency. Low intensity or light stimulus, just a few action potentials per second. Really intense, maybe more than 100 action potentials per second. Connor? How are afferent neurons or pathways functionally different from efferent neurons? What types of information do they send is what I'm trying to ask here. Yeah, so two motor output that's going to cause muscle contraction, and then sensory information that's going to relay information about our body and our environment back. Afferent neurons carry sensory information, which is really what I want you to know from this slide. Afferent neurons carry sensory information. Efferent neurons are also known as motor neurons, and they're going to cause muscle contraction. So the electrical signals flow in different ways. Afferent neurons, signals flow up the spinal cord back to the brain. Efferent flow away from the brain out to the muscles. Name 6 muscles that are agonists for flexion at the tibiofemoral joint, so at the knee. Okay, good. Sartorius. Your hamstrings, yep. So what do you remember the hamstring names? Quad. Okay. So you got sartorius for sure. You got the hamstring muscles. So you got semimembranosus, semitendinosus, and biceps femoris. Remember, that's medial to lateral. All right, so there's 4. What are some other? big one. So gastroc, plantaris, and then we said sartorius, but the other pes and serine muscles. So semitendinosus we already counted, but gracilis is another one. So there's a bunch of little knee flexors. All right, getting down to the end of this board. Fundamentals for 3. At resting membrane potential, why does a small amount of sodium leak into a cell rather than leaking out of the cell? At rest, some sodium does leak inside of it. Why does sodium leak inside rather than going the other way? It'll actually make the inside of the cell more positive because it's a positive ion coming inside. The answer is, and this is one of those things I didn't know how well I'd explain, so I want to make sure I put it on the slideshow. So the answer is it diffuses along its concentration gradient. Area of high, lots of sodium, and then it's going to go from concentration to not much sodium inside of the cell at rest. And so given its druthers, given what it wants to do, it'll flow with its concentration gradient from high concentration to low, which means it's from outside of the cell to inside if it's given the opportunity. All right. Describe chemically gated and voltage gated channels and the role that each plays in generating an action potential. So yeah, once it hits negative, so it's going to move from negative 70 up to negative 55, and that's the threshold voltage that then opens the voltage gauge. Yep. Yeah. So yeah, good. So chemically, remember, whatever the first thing is the key to open that door. So chemically, to receipt of a neurotransmitter. So something like glutamate. So a really common example in the central nervous system. And so in response to receiving glutamate, chemically gated channels are going to open. That's going to cause a bunch of sodium to come rushing inside of the neuron. And as sodium comes rushing inside, that part of the neuron gets more positive. And once we hit that threshold, then the channels open. And then that wave of depolarization, known as an action potential, that goes from the dendrites through the cell body and down the axon out to the terminals. So chemically gated channels open first, and then they cause the opening of voltage gated channels under normal circumstances. Unless you're applying an electrical current to yourself. All right, so back to Connor. All right, how are type 1 and type 2 muscle fibers different? So let's go type 1 versus. Yep. Okay, good. Those are pretty much the bullet points. Sometimes they are primarily can't produce much, but they can produce a little bit of force for a long time, hours on end. As opposed to your type 2X fibers are larger, so they can produce quite a bit more force, but they fatigue really quickly. They fatigue within a couple of minutes. And so then the type 2As are kind of an intermediate. So they have some oxidative capacity, some anaerobic capacity. They fatigue faster than the type, not quite as fast as the type 2Xs. All right, going for 5. All right, so starting with release of calcium from the sarcoplasmic reticulum, what are the events of cross-bridge formation? The sarcoplasmic reticulum is going to the voltage. The sarcoplasmic is going to cause the release of calcium, which is our starting point here. So calcium then moves down inside of the myofibril, and it's going to P down. Otherwise, calcium gets pumped back into the sarcoplasmic reticulum. Troponin C goes back to its original shape and covers the binding sites back up. And then 4. All right, so from a neuromuscular standpoint, describe 2 ways to increase the amount of tension generated by a muscle. So the 2 broad ways to do this are motor unit recruitment and rate coding. Remember that motor unit recruitment, or backing up a step, remember that a motor unit is an alpha motor neuron, which is a little neuron body is in the spinal that axon runs out to the muscle fibers. We'll be talking about the biceps, flexor. That alpha motor neuron and all the fibers that it innervates. Remember, big motor units, maybe a few thousand fibers, so we get big contractions. Small motor units, like we get small contractions, but they're much finer. So what we're going to do then is recruit motor units based on the size principle. We start with the small ones, which are type 1 motor units, and work our way up to the big ones. So that's motor unit recruitment. If you recruit all the type 1s, it's not enough to lift whatever you're trying to lift. Then you recruit the 2A's, then the 2X's. Rate coding is the number of times per second that the alpha motor neuron depolarizes. So it's in terms of grading the intensity of the stimulus. So we want action, alpha motor neuron sends more action potential per second, so greater frequency. All right, we got about 10 minutes left, so then we can knock out these 2 columns. So we got potpourri in the arm. Okay. List the 8 carpal bones. Really? Okay. They're all in here somewhere. All right. So you're on a roll, though. That was good. Yep, capitulum. Or not, the capitellum, I'm sorry. Okay. So the one that we're missing I know of is the pisiform. And then triquetrum. So the thumb one. I think you said trapezoid. Maybe you said that one too. Okay, good. All right, and that was 100 points. Connor, what do you think? The central nervous system includes, so what are the 2 components of the central nervous system? Really? Brain and spinal cord. So yeah, central nervous system, brain and spinal cord, and then peripheral nervous system, everything else. So central nervous system is only the brain and spinal cord. All right, back to, yep, there we go. List 5 agonists for radiocarpal flexion. Remember, your radiocarpal joint's your wrist joint. Well, there can only be 5 forearm questions, so I guess that's some good news. Connor, you got anything offhand? Wrist flexors? All right, so the wrist flexors, remember one of the groups, the superficial group, is where you take one hand, put it over the other elbow. We're going to, yeah, so we're going to ignore my thumb for now. But you got flexor carpi radialis, palmaris longus, flexor carpi ulnaris, so there's 3. Flexor digitorum superficialis, flexor digitorum profundus, those are kind of the big 5. You can throw flexor pollicis longus in there if you want. So yeah. Connor, what are we going with? Potpourri for 5? All right, name 2 joints in the upper extremities, or upper extremity, that have 2 degrees of freedom. So remember, 2 degrees of freedom means that it moves in 2 planes, and only 2 planes. So that's a joint, or one plane. Yeah, this is a tough question. All right, so I can think of 3 offhand. What's that? Radiocarpal is one. Yep. So radiocarpal joint, you can plane. 1 degrees of freedom. The other 2 I can think of are in the hand, or 2 types. So you have the metacarpophalangeal in digits 2 through 5. So your knuckle. All right. How are the sympathetic and parasympathetic branches of the autonomic nervous system? Which one's fight or flight? Okay. Sympathetic, yeah. So sympathetic's fight or flight. So remember, that's going to do things like increase your heart rate, increase your breathing rate, pupil dilation. It's going to start to free up glucose so you for exercise. Yeah. It's the opposite. So parasympathetic branch is going to cause you to direct blood flow toward the abdomen. So you're going to start to filter blood in the liver, or more exercise, you're going to filter blood. Direct blood flow to the stomach, to the intestines, going to break down. So those are the things that the parasympathetic does. So sympathetic, fight or flight, all things we need to do either of those. And then parasympathetic, rest and digest. So slows your heart rate down and then activates the digestive system. All right. Connor? List 6 actions that occur at the glenohumeral joint and the plane and axis of each. Correct. Yep. So good. So you can do also ABA deduction, which is in the frontal plane, the z-axis, and the transverse or horizontal plane, and the y-axis. All right. List 5 agonists for glenohumeral flexion. So remember the glenohumeral joint. To bring the arm forward, you've got muscles like pec major, anterior delt, coracobrachialis, and then I meant to write extension. So all the agonists. For extension. Posterior delt or rear delt is another one. What else we got? And then I think it's a bad question. What are some other ones? What else is an extensor? What's that? No, because that's a scapular muscle. Yeah, that was a bad question. Yeah, so there's 4 that I can think of offhand for that one too. So posterior delt, triceps, teres major, and then latissimus dorsi are the big ones there. Yeah. All right, well, I should stop being distracted while I'm writing Jeopardy questions. All right, which of the three, Connor? Let's go. All right, why are astrocytes crucial for normal central nervous system function? Okay, so there's a category. So they provide scaffolding for the neuron, and then that they help. So that memory is important for the storage of long-term memory, so they can physically make those connections between the neurons. They clean up the electrical around glutamate from that synapse to neurons. And then they also regulate blood flow to the neurons. So with those feet of the astrocytes, they can relax them to allow more blood flow, or they can close them down to constrict and direct blood flow elsewhere to other neurons that are doing more stuff. All right, 200s. We named 3 extrinsic muscles of the thumb. There's 4 of those that I can think of. Yeah, there are 4 of them. Anybody? So extrinsic means they originate somewhere in the forearm and then they insert somewhere in the thumb. As opposed to, like, you have thumb muscles here in this area, that fat part of your hand called the thenar eminence. There's some thumb muscles in there. So not those, but the ones that originate in the forearm. So the thumb is, in the muscles, it's called pollicis. Okay. So the muscles are, remember, so you got your anatomical snuff box. So you've got the extensor pollicis longus. Extensors and last, potpourri for 2. Maybe. There we go. Describe microglial cells. Because we're out of time, those are the brain's immune cells. So remember that because of that blood-brain barrier, some of the big cells of your immune system can't cross that barrier, so the brain has to have its own immune cells, and so those are the microglial cells. So the brain's own immune cells are what those things are. Cool. So like I said, I'll post this Word document in Canvas, and it has the links to the game we just played, and now you have the answers, and then other things to know down on that. So, and then like I said, test on Monday, so I won't see you on Monday, but I'll see you again on Wednesday, and we'll start the ankle and lower leg. So we'll see you all then.